अस्सलाम वालेकुम स्टूडेंट्स। होप यू all are fine। स्टूडेंट्स आज हम बायो क्लास 11 चैप्टर नंबर 6 एक्सरसाइज शॉर्ट क्वेश्चन आंसर्स सॉल्व करेंगे। सो लेट्स स्टार्ट। फर्स्ट क्वेश्चन है हमारे पास, राइट द पिगमेंट कंपोजिशन ऑफ साइनोबैक्टीरिया। आंसर इज साइनोबैक्टीरिया also known as blue-green algae have a unique pigment composition that allows them to photosynthesize and thrive in various environments. The main pigments found in cyanobacteria are: first one is chlorophyll A. This green pigment is responsible for absorbing light energy and plays a central role in photosynthesis. Next is phycocyanin. This blue pigment is a bilin protein that absorbs red light and scatters blue light, giving cyanobacteria their characteristic blue-green color. Allophycocyanin. This pigment is a variant of phycocyanin and also absorbs red light. Next is carotenoids. These yellow-orange pigments such as beta-carotene and zeaxanthin absorb blue and red light and protect the cells from excessive light energy. Next is phycoerythrin. This red pigment is present in some cyanobacteria and absorbs green light. The combination of these pigments allows cyanobacteria to absorb a wide range of light wavelengths, making them efficient photosynthesizers in various aquatic environments. Next question is, do you know the difference between bacteria and archaea? Bacteria and archaea are both microorganisms, but they belong to distinct domains of life. Here are the main differences. First one is cell wall. Bacterial cell wall is made up of peptidoglycan, also known as murine. Archaea lack peptidoglycan. Instead, they have pseudopeptidoglycan, glycoproteins, or polysaccharides. Next is membrane lipids. Bacteria esters in their membrane lipids, archaea ethers in their membrane lipids. Next is metabolism. Bacteria more diverse metabolic processes including photosynthesis, respiration, fermentation. Archaea unique metabolic processes such as methanogenesis, producing methane gas, and halophily, thriving in high salt environments. Next is reproduction. Bacteria typically reproduce by binary fission. Archaea reproduce by binary fission, budding, or fragmentation. Next is genetics. Bacteria have a single circular chromosome. Archaea have a single circular chromosome, but some have multiple chromosomes. Next is environmental adaptations. Bacteria found in various environments including soil, water, air, and living organisms. Archaea often found in extreme environments such as hot spring, salt lake, and deep sea vents. Evolutionary history. Bacteria diverged from a common ancestor around 3.5 billion years ago. Archaea diverged from a common ancestor around 3.5 billion years ago, but their lineage is thought to be older than bacteria. These differences reflect distinct evolutionary paths and adaptations to various environments. While both domains share some similarities, their unique characteristics set them apart. Next question is, What are the morphological forms of bacteria? Answer is, bacteria exhibit a wide range of morphological forms including spherical, cocci, round or oval shape, for example Staphylococcus. Next are rod-shaped, which are called bacilli. These are straight or curved rods, for example Escherichia coli. Next is spiral, spirilla, helical or spiral in shape, for example Helicobacter pylori. Next are comma-shaped Vibrio, curved rods with a comma-like shape, for example Vibrio cholerae. Next is pleomorphic, irregular shapes, for example Mycoplasma. Filamentous, long, thin filaments, for example actinomyces. Diplococci, pairs of spherical cells, for example Streptococcus pneumoniae. Streptobacilli, chains of rod-shaped cells, for example Streptobacillus moniliformis. Coccobacilli, short rod-shaped cells with a slightly rounded end, for example Haemophilus influenzae. These morphological forms are often used in combination with other characteristics such as staining properties and metabolic features to identify and classify bacteria. Next question is, give the functions of following in bacteria. First one is ribosomes, cell membrane, nucleoid, plasmid, mesosomes, slime capsule, flagella, cell wall, pili. First one is ribosomes, site of protein synthesis responsible for translating messenger RNA into specific sequence of amino acid. Next is cell membrane, which is also called plasma membrane, regulates the movement of material in and out of the cell, maintains cellular homeostasis, generates energy through cellular respiration. Next is nucleoid, contains bacterial chromosome, genetic material, site of DNA replication and transcription. Plasmid, small, self-replicating, circular DNA molecules, carry genes for specialized functions, for example antibiotic resistance. Mesosomes, enfoldings of cell membrane, increase surface area for cellular respiration and photosynthesis. Next is slime capsule or glycocalyx, protective layer outside the cell wall, helps bacteria adhere to surfaces and resist phagocytosis. Next is flagella. Long whip-like structures provide motility and movement. Next is cell wall. Maintains cellular shape and structure, protects against osmotic pressure and environmental stress. Next is pillars or fimbriae. Short hair-like structures aid in attachment to surfaces and DNA transfer conjugation. These components work together to enable bacteria to survive, reproduce, and interact with their environment. Next question is, how the mechanism of photosynthesis in cyanobacteria is similar and different from that of plant. Answer is, aquatic cyanobacteria and terrestrial plants share similarities in photosynthesis but also exhibit differences due to their distinct environments and adaptations. Here's a comparison. First of all, we see similarities. Light absorption. Both use pigments, chlorophyll A, phycocyanin, and carotenoids to absorb light energy. Light-dependent reactions. Both generate ATP and NADPH through light-driven electron transport. Calvin cycle. Both use Calvin cycle or C3 pathway to fix carbon dioxide into organic compounds. Oxygen evolution. Both produce oxygen as a byproduct of photosynthesis. Next is differences. Pigment composition. Cyanobacteria have phycocyanin and allophycocyanin. while plants have chlorophyll B and other accessory pigments. Light harvesting complexes. Cyanobacteria have phycobilisomes, while plants have light harvesting complexes LSCs associated with photosystems. Photosynthetic units. Cyanobacteria have smaller photosynthetic units, photosystems compared to plants. Electron transport chain. Cyanobacteria have a more flexible electron transport chain, allowing for adaptation to changing light conditions. Carbon dioxide concentration mechanisms. Cyanobacteria have carbon dioxide concentrating mechanisms to cope with low aquatic carbon dioxide levels, while plants have stomata to regulate CO2 uptake. Next is water source. Cyanobacteria use water from their aquatic environment, while plants use water from the soil. Gas exchange. Cyanobacteria exchange gases directly with water, while plants exchange gases through stomata. These differences reflect adaptations to their respective environments, allowing cyanobacteria to thrive in aquatic ecosystems and plants to dominate terrestrial ecosystems. Next question is draw and label structure of flagellum. कि फ्लैजेलम को स्ट्रक्चर को ड्रा करें और लेबल करें। तो ये है बच्चों, ये पीछे चैप्टर में भी दिया हुआ है। हमने पढ़ा था कि फ्लैजेला के 3 पार्ट्स होते हैं, एक बेसल बॉडी होती है, एक हुक होता है और एक फिलामेंट होता है। ठीक है, तो ये देखिए, ये आपने ड्रा करना है और लेबल करना है इन पार्ट्स को। नेक्स्ट क्वेश्चन इज हाउ कीमोसिंथेटिक बैक्टीरिया आर ऑटोट्रॉफिक इन नेचर। कीमोसिंथेटिक हमने बच्चों जब मोड्स ऑफ न्यूट्रिशन ऑफ बैक्टीरिया पढ़े थे तो उसमें हमने 2 पढ़े थे, ऑटोट्रॉफिक मूड भी होता है, हिटोट्रॉफिक भी। और जो हमारे पास ऑटोट्रॉफिक था उसमें भी हमने फर्दर 2 ग्रुप्स पढ़े थे, एक फोटोसिंथेटिक होते हैं और एक कीमोसिंथेटिक बैक्टीरिया होते हैं। ठीक है, so chemosynthetic bacteria are autotrophic in nature because they produce their own food using chemical energy rather than relying on sunlight or organic compounds. कह रहा है कि कीमोसिंथेटिक बैक्टीरिया जो है ना वो ऑटोट्रॉफिक ही होते हैं नेचर में। कह रहा है जो कीमोसिंथेटिक बैक्टीरिया है वो अपनी जो फूड है वो खुद प्रिपेयर करते हैं केमिकल एनर्जी को यूज करते हुए रेदर देन डिपेंडिंग ऑन सनलाइट और ऑर्गेनिक कंपाउंड्स। हियर्स हाउ chemical energy source. They use inorganic compounds like hydrogen, gas, sulfur, iron or ammonia as energy source. Oxidation reactions. They oxidize these organic compounds, releasing energy. ATP synthesis. This energy is used to generate ATP through chemiosmosis, similar to mitochondrial ATP synthesis. Next is carbon fixation. They use ATP to fix carbon dioxide into organic compounds such as glucose through Calvin cycle or other carbon fixation pathways. Next is organic compounds synthesis. They synthesize their own organic compounds like sugars, amino acids, and nucleotides using the fixed carbon. By using chemical energy to produce their own food, chemosynthetic bacteria are autotrophic, meaning they don't rely on sunlight. Photoautotrophy, don't consume organic compounds. Heterotrophy, produce their own food using chemical energy. Chemoautotrophy, examples of chemosynthetic bacteria include hydrogen oxidizing bacteria, for example Equifax; sulfur oxidizing bacteria, for example Thiobacillus; iron oxidizing bacteria, for example Ferrobacillus; ammonia oxidizing bacteria, for example Nitrosomonas. These bacteria play crucial roles in ecosystem, contributing to nutrient cycling, primary production, and supporting life in diverse environments. Next question is which chemical methods are used to control microbes? बच्चा हमने बहुत सारे मेथड्स पढ़े थे, केमिकल मेथड्स माइक्रोब्स को कंट्रोल करने के लिए, लाइक डिसइंफेक्टेंट्स, एंटीसेप्टिक्स, वो सारे आपने बताने हैं। Several chemical methods are used to control microbes including disinfectants, chlorine, iodine, and quaternary ammonium compounds, quartz, kill or inactivate microbes on surfaces. Next is antiseptics, hydrogen peroxide, ethanol, and povidone reduce microbial growth on living tissues. Preservatives, say sodium benzoate, potassium sorbate, and calcium propionate prevent microbial growth in foods and pharmaceuticals. Sterilants, ethylene oxide, formaldehyde, and glutaraldehyde kill all microbes, including spores, for sterilization purposes. Next is antibiotics. Target specific microbial metabolic processes, inhibiting growth or killing microbes. Heavy metals. Copper, silver, and mercury have antimicrobial properties used in medical devices and water treatment. Acidulant: acetic acid, lactic acid, and citric acid create acidic environments, inhibiting microbial growth. Oxidizing agents: hydrogen peroxide, ozone, and chlorine dioxide kill microbes through oxidation. Aldehydes: formaldehyde, glutaraldehyde, and glyoxal have antimicrobial properties used in disinfectants and sterilant. Phenolics: phenol, cresol, and hexachlorophene disrupt microbial cell membranes used in disinfectants and antiseptics. These chemical methods are used in various applications including medicine, food industry, water treatment, and surface disinfection to control microbial growth and prevent infections. Next question is, give physical methods to control microbe. First one is heat. Moist heat, autoclaving, boiling; dry heat, incineration, hot air sterilization; radiation, ultraviolet light, X-rays, gamma rays, electron beams; filtration, membrane filters, microfiltration, ultrafiltration; depth filters, activated carbon, diatomaceous earth; desiccation or drying; dehydration, freeze drying; lyophilization; high pressure, high hydrostatic pressure (HHP), high pressure processing (HPP); ultrasound, high frequency sound waves, ultrasonic cleaning; microbial traps, HEPA filters, high efficiency particulate air filters; electrostatic precipitation; cold temperatures, refrigeration, freezing, vacuum; vacuum drying, vacuum packaging; physical removal, washing, rinsing, scrubbing. These physical methods are used to control microbial growth, reduce microbial loads, or sterilize surfaces, equipment, and materials in various industries including healthcare, food processing, and pharmaceuticals. Next question is, name any two bacteria that cause diseases in plants. First one is Agrobacterium tumefaciens, causes crown gall disease in plants leading to tumor-like growths on stem and root. Next is Pseudomonas shrinch, causes various diseases including bacterial leaf spot and blight, bacterial canker, bacterial speck. These bacteria can infect a wide range of plant species, including crops and ornamental plants, causing significant economic losses and impacting agricultural productivity. Next question is, name any 5 diseases caused by bacteria in man. First one is tuberculosis, TB, caused by Mycobacterium tuberculosis, affecting lungs and potentially other organs. Pneumonia, caused by Streptococcus pneumoniae, Klebsiella pneumoniae, or other bacteria infecting lungs. Typhoid fever, caused by Salmonella typhi, affecting digestive system and bloodstream. Diphtheria, caused by Corynebacterium diphtheria, affecting throat and potentially heart and nervous system. Leprosy, Hansen's disease, caused by Mycobacterium leprae, affecting skin, nerves, and mucous membrane. These bacterial diseases can have significant impact on human health, and some like TB and typhoid fever remain major public health concerns globally. Next question is, define the term normal flora. Normal flora, also known as commensal flora, refers to community of microorganisms like bacteria, viruses, fungi, and protozoa that naturally inhabit human bodies, skin, mucous membrane, gastrointestinal tract, respiratory tract, etc. Coexist peacefully with their host like humans, do not cause diseases under normal circumstances, perform beneficial functions such as aiding digestion and nutrient absorption, producing vitamins, for example vitamin K, regulating immune system, preventing colonization by pathogenic microorganisms. Examples of normal flora include skin, Staphylococcus epidermidis, Cyanobacterium species, gut, Escherichia coli, E. coli, Lactobacillus species, Bifidobacterium species, respiratory tract में हमारे पास बैक्टीरिया जो होते हैं सेप्टोकोकस नमूनी, हीमोफिलस इन्फ्लुएंजा, Vagina, Lactobacillus acidophilus. Normal flora plays a vital role in maintaining human health and preventing infections. Disruptions to the balance of normal flora, also known as dysbiosis, can lead to various health issues. Next question is, what is chemical composition of cell wall of bacteria? The chemical composition of the bacterial cell wall varies depending on the type of bacteria, but most bacterial cell walls contain peptidoglycan, also known as murine, a polymer of sugars N-acetyl-glucosamine and N-acetylmuramic acid cross-linked by short peptides, provides structure support and maintains cell shape. Teichoic acid, polymers of glycerol or ribitol phosphate linked to peptidoglycan, found in gram-positive bacteria, helps maintain cell wall stability. Next is lipoic acid, a molecule composed of a glycerol phosphate backbone linked to peptidoglycan, found in gram-positive bacteria, helps anchor the cell wall to membrane. Lipopolysaccharides, also known as endotoxins, a complex molecule consisting of a lipid, a component, a core polysaccharide, and O-antigen chains, found in gram-negative bacteria, helps maintain outer membrane stability. Next is proteins. Various proteins such as porins, enzymes, and structural proteins are embedded in or attached to the cell wall. Other components. Some bacteria have additional cell wall components like mycolic acid in Mycobacteria or pseudopeptidoglycan in Archaea. The exact composition and arrangement of these components vary between different bacterial species and even within the same species depending on factors like growth condition and environmental pressures. Next question is, distinguish between lysosome and mesosome, peptidoglycan and muramic acid, gram-positive and gram-negative bacteria, lytic and lysogenic bacteria, pathogenic and non-pathogenic bacteria, autotrophy and heterotrophy, photosynthetic and chemosynthetic bacteria, mutation and mutant, chromosomes and bacteriophage, bacteria and mitochondria, prokaryotes and eukaryotes, cyanobacteria and bacteria. So first of all, we see difference between lysosome and mesosome. Lysosomes, eukaryotic organelles containing digestive enzyme. Mesosomes, bacterial infoldings of cell membrane. Peptidoglycan and muramic acid. Peptidoglycan, polymer of sugars and amino acids in bacterial cell wall. Muramic acid, component sugar of peptidoglycan. Gram-positive and gram-negative bacteria. Gram-positive, thick peptidoglycan layer, teichoic acids and lipoteichoic acids. Gram-negative, thin peptidoglycan layer, outer membrane with lipopolysaccharides. Next is difference between lytic and lysogenic bacteria. Lytic, viruses that immediately infect and lyse host cells. Lysogenic, viruses that integrate into host DNA remaining dormant. Pathogenic and non-pathogenic bacteria. Pathogenic, bacteria causing disease. Non-pathogenic, bacteria harmless to humans. Autotrophy and heterotrophy. Autotrophy, organisms producing their own food. Heterotrophy, organisms consuming external food sources. Next is photosynthetic and chemosynthetic bacteria. Photosynthetic bacteria using light energy, chemosynthetic bacteria using chemical energy. Mutation and mutant. Mutation change in DNA sequence, mutant organism with a muted trait. Chromosome and bacteriophage. Chromosome DNA containing structure in cell. Bacteriophage viruses which infect bacteria are called bacteriophage. Next is bacteria and mitochondria. Bacteria, prokaryotic microorganisms, mitochondria, eukaryotic organelle responsible for energy production. Prokaryotes and eukaryotes. Prokaryotes, cells lacking a true nucleus. Eukaryotes, cells with a true nucleus. Cyanobacteria and bacteria. Cyanobacteria are photosynthetic bacteria. Bacteria broad term encompassing many types including cyanobacteria. Next is, what are plasmids? Plasmids are small self-replicating circular DNA molecules found in bacteria and other microorganisms. They are independent of chromosomal DNA. Plasmids replicate separately, usually carry non-essential genes, but may provide beneficial traits like antibiotic resistance, toxin production, virulence factors, metabolic pathways. Can be transferred between cells through conjugation, direct cell-to-cell transfer, transformation, uptake of free DNA, transduction, virus-mediated transfer. Used in genetic engineering as vectors to introduce genes into cells. Plasmids are important tools in molecular biology and biotechnology, allowing scientists to study gene function and regulation, develop new genetic traits in microorganisms, produce recombinant proteins and other valuable compounds. Plasmids can be classified into different types based on their characteristics such as conjugative plasmids can transfer themselves between cells, non-conjugative plasmids cannot transfer themselves, cryptic plasmids have unknown functions, virulence plasmids carries genes related to pathogenicity, plasmids play a significant role in microbial genetics, evolution and biotechnology. Next question is, how do bacteria survive under unfavorable conditions? Bacteria have evolved various strategies to survive under unfavorable conditions, including dormancy, entering a dormant state such as endospores to withstand extreme temperatures, radiation, and chemicals. Biofilms, forming complex communities attached to surfaces, providing protection against antimicrobials and environmental stresses. Sporulation, producing highly resistant endospores that can survive extreme conditions. Adaptation, modifying their physiology and metabolism to cope with changing environments. Genetic variation, utilizing genetic mechanisms like mutation, recombination, and horizontal gene transfer to adapt to new conditions. Quorum sensing, communicating with each other to coordinate behaviors and optimize survival. Antibiotic resistance, developing mechanisms to resist antibiotic treatment, stress responses, activating specific pathways to mitigate stressors like heat shock, oxidative stress, or nutrient deprivation. Dwarf formation, entering a state of reduced metabolic activity to conserve energy. Cryptobiosis, entering a state of suspended animation where metabolic processes come to a near halt. These strategies enable bacteria to survive and even thrive under unfavorable conditions, making them incredibly resilient microorganisms. Next question is, list 5 ways in which bacteria are beneficial to man. First one is gut health and digestion. Beneficial bacteria in the gut like lactobacillus and bifidobacterium aid digestion, produce vitamins, and regulate immune system, fermentation and food production. Bacteria like Lactococcus and Streptococcus are used in fermenting foods like yogurt, cheese, sauerkraut, bread, creating nutritious and flavorful products. Medicine and antibiotics. Bacteria like Streptomyces and Bacillus produce antibiotics such as streptomycin and bacteriacin used to treat various infections. Environmental cleanup. Bacteria like Pseudomonas and dihalococcaides can degrade pollutants including oil spills, pesticides, and industrial contaminants, helping to clean the environment. Vaccine development and immune system support. Beneficial bacteria like Bacillus calmette-guerin (BCG) are used in vaccines to boost the immune system and protect against diseases like tuberculosis. These examples illustrate the significant contributions of bacteria to human health, food production, medicine, environmental sustainability, and more. Next question is, why are cyanobacteria considered as the most prominent of photosynthetic bacteria? Cyanobacteria are considered the most prominent photosynthetic bacteria for several reasons. First one is oxygenic photosynthesis. Cyanobacteria are only bacteria capable of oxygenic photosynthesis, producing oxygen as byproduct like plants and algae. Evolutionary significance. They are thought to have played a crucial role in oxygenating the Earth's atmosphere around 2.7 billion years ago. Next is diverse habitats. Cyanobacteria thrive in various environments including freshwater, marine, terrestrial, extreme habitats like hot springs and Antarctic ice. Next is nitrogen fixation. Many cyanobacteria can fix atmospheric nitrogen, making it available to other organisms. Primary producers. They are primary producers in many ecosystems, forming the base of aquatic food webs. Symbiotic relationship. Cyanobacteria have symbiotic relationship with fungi like plants, for example azolla, and animals, for example corals. Ecological importance. They contribute to nutrient cycling, carbon sequestration, and soil formation. Biotechnological applications. Cyanobacteria are used in biotechnology for producing biofuels, chemicals, and pharmaceuticals. Genomic diversity. They have diverse genomes providing insights into evolutionary processes and adaptations. Next is ancient lineage. Cyanobacteria have an ancient lineage with fossil evidence dating back over 3.5 billion years. These factors combined make cyanobacteria the most prominent photosynthetic bacteria with significant impacts on the environment, ecology, and human society. Next question is, what are the benefits of bacterial flora to human? Bacterial flora, also known as human microbiome, provides numerous benefits to humans including digestion and nutrient absorption. Bacteria help break down complex foods, absorb nutrients, and produce vitamins. Immune system development and function. Bacteria aid in the development and regulation of the immune system. Pathogen protection. Beneficial bacteria can outcompete harmful pathogens, reducing the risk of infection. Production of antibiotics and antimicrobials. Certain bacteria produce compounds that help fight off harmful microorganisms. Next is modulation of inflammation. Bacteria can influence inflammation levels, reducing the risk of chronic diseases. Maintenance of gut barrier function. Bacteria help maintain the integrity of the gut lining, preventing leaky gut syndrome. Influence on brain function and behavior. The gut-brain axis suggests that bacteria can impact mood, cognitive function, and behavior. hormone regulation. Bacteria can influence hormone production and balance. Cancer prevention. Certain bacteria may help prevent cancer by promoting a healthy gut microbiome. Improved mental health. Bacteria can produce neurotransmitters and influence mental well-being. Next is enhanced detoxification. Bacteria can aid in removal of toxins from body. Support for gut-associated lymphoid tissue galled. Bacteria help maintain the health and function of gall. These benefits highlight the importance of maintaining a balanced and diverse gut microbiome for overall human health and well-being. Next question is, what is the primary function of the F factor in bacterial cells? The primary function of the F factor, fertility factor, in bacterial cells is to facilitate the process of conjugation, which is a type of horizontal gene transfer between bacteria. The F factor is a plasmid that carries genes responsible for conjugation. The F factor encodes proteins that form a pillar which connects two bacterial cells and enables the transfer of DNA. DNA transfer. The F factors allows the transfer of genetic material, plasmid or chromosomal DNA from a donor cell F positive to a recipient cell F negative. Replication. The F factor can replicate independently of the bacterial chromosome, ensuring its maintenance in the population. Next is stability. The F factor can integrate into the host chromosome, becoming a stable component of bacterial genome. The F factor plays a crucial role in bacterial evolution, allowing for gene sharing, exchange of genetic material between bacteria, promoting genetic diversity, adaptation, acquisition of new traits such as antibiotic resistance or virulence factors, evolution, facilitating the spread of beneficial genes through bacterial populations. In summary, the F factor is a critical component of bacterial conjugation, enabling the transfer of genetic material and promoting genetic diversity, adaptation and evolution in bacterial populations. So students, हमारे ये क्वेश्चंस कंप्लीट हो चुके हैं. होपफुली आपको समझ आ गए होंगे. इफ यू हैव एनी डाउट यू कैन आस्क इन कमेंट सेक्शन. प्लीज लाइक शेयर एंड सब्सक्राइब माय यूट्यूब चैनल. थैंक यू.